Bismillahirrahmanirrahim. Dear students, have a nice day. Hope you will be fine best of your health. This is Dr. Sajid Mahmood Rao, Division of Science and Technology, University of Education, Lahore. Today, we are talking about steroidal hormones, its synthesis and pharmaceutical applications, as well as steroidogenesis. The key concepts which we are going to discuss in this class session are steroid hormones, its sources, steroidogenesis, hormone synthesis, and their functions. Let's see the history of hormones. Hormones is a Greek word means arouse, that is excite, a chemical substance produced in the body that controls and regulates the activity of certain cells or organs. Many hormones are secreted by special glands, such as thyroid hormones produced by the thyroid glands. When we see the history of hormones, you can see over here the first hormone that was synthesized and discovered by Baylis and Starling in 1902. They discovered the first GIT hormones, and the name of that hormone is secretin. After that, different authors define hormones in their own ways. Grigore and Tracy in 1964 defined the chemical definition of gastrin. After that, Jallo and Benson in 1966 invented RIA to estimate the quantity of hormones. Jobs and Mute in 1996 explained the chemical nature of secretins and cholecystokin and a group of other hormones. In vertebrates, steroids acts on a wide range of tissues and influence many aspects of biological including sexual differentiations, reproductive physiology, osmoregulations and intermediate metabolism. Major sites of steroid synthesis and secretions include the ovaries, testes, adrenal, and placenta. Based on the distance of the target site from the site of synthesis and secretions, steroid hormones can be classified as either endocrine distant target tissues, paracrine neighboring cells, or autocrine that is called same cells factors. When secreted into the environment, steroids can also act as pheromones by conveying information to other organisms. Owing to the pervasive effects of steroids in vertebrates biology, a number of pathologic states can occur because of problems related to steroid hormones action. These diseases states include cancer, steroid insensitivity, and abnormal steroid synthesis. The purpose of this chapter is to provide an overview of steroid synthesis, steroid hormone effects in normal physiology, molecular and biochemical mechanism of action of steroid hormones, and pathologic states related to the steroid hormones action. Let's see what is steroid hormones. However, before going to discuss the basic concepts of the steroid hormones, here I will let you know the general overview of the steroids. Steroids are the lipids. They are soluble in organic solvents. Steroids include wide range of naturally occurring compounds like sterols, cholesterols, and bioacids. Steroid hormones basically produced in the adrenal cortex, testes, ovary, and some peripheral tissues, that is, adipose tissues and the brain. All steroid hormones share a typical but not identical in their ring structures. All steroid hormones are derived from cholesterol and differ only in the ring structures and side chains attached to it. Generally, the steroids hormones are soluble in organic solvents and they are found as lipids in nature. You can see over here the basic structures of pregnenolone, in which you can observe the presence of three rings which are hexagonal and the fourth ring is pentagonal. The hydroxyl group is present at carbon 3 position while double bond is appeared at carbon 5 position. The acetyl group is appeared at carbon 17 position. So the total number of carbon atoms in the pregnenolone structures is 21. When we see the basic sources of hormones, they are derived from milk and milk products, specifically the dairy fat in butter and cheese. They provides a very small amount of progesterones. This is the hormone that predominates in the first half of a woman's menstrual cycle, and that supports the growth of the unborn child during pregnancy. It also relaxes the smooth muscles, such as those surrounding the bronchial passages, normalizes blood clotting, reduces gallbladder activity, and encourages the immune response. Hormones are also isolated from eggs, meat, and fish that contain small amount of estrogens and testosterones identical to their human form. Eggs providing approximately 10 to 20% as much as meat and fish. Plant foods have not been found to supply actual human estrogens, which is not the same as the phytoestrogens. Phytoestrogens are those which are isolated and derived from the plants. But some plants food contain minuscule amount of testosterone, that is the natural hormone sources. Let's see the isolation and purifications of steroids. This is the specific method for the extraction and isolation of steroid molecules. In this method, we will take hot boiled egg yolk in which 1 gram of potassium carbonate is added. After that, 5 gram of sand and 10 ml of methanol is added. Whole material is grinded and we will get the soft scrambled egg material. In this material, we will add 20 ml of cyclohexane and stir it for some times. After that, we will take the cornmeal mush material. The whole material is then formed for reflex and evaporation. After evaporations, we will get the evaporated egg mixtures in which we will add 30 ml of methylene chloride. Now, this material is subjected for column chromatography in which we will take 15 gram of flash silica gel and layer of sand on the top. So, the column is packed. After that, the whole column is eluted with 30 ml of methylene chloride. Now, it is rinsed with ethanol with the help of petroleum ether. After that, again, the elution is carried out with the help of ethanol and petroleum ether with the specific ratio. So, in this way, from the column chromatography, we will get total 15 fractions and each fractions have 10 ml of volumes. Now, each fraction is subjected for thin layer chromatography for the identifications of the pure compound as well as for the presence of steroid molecules. So, the cholesterol will be spread into 6 to 16 fractions. Now, after the evaporations of cholesterol fractions, we will get the cholesterol crystals in the round bottom flask. Now, the steroid, which is in the form of cholesterol, is recorded for weight and then taken its melting point. After that, the cholesterol is identified with the help of specific test for the conformations of the steroid molecules. So, this is the specific method for the isolations and purifications and identifications of the steroids natural products. Let's see what is steroidogenesis. After taking this lecture, you should be able to describe the key components of steroidal genesis, list the major steroid hormone classes and key examples, the locations of where steroid hormone synthesis occurs, and the detailed steps of making steroid hormones from cholesterol. When we see the statement of steroidogenesis, you can call that the steroidogenesis is the biological process by which steroids are generated from cholesterol and changed into other steroids. The pathway of steroidogenesis differ among species. So, let's start with the steroid hormone classes, and we can see we have one, 2, 3, 4, and 5 major classes of steroid hormones. Now, each of these hormones classes, the way to think about it is that they are defined by the receptors that they activates, which then dictates the physiological effects of these hormones classes. So we start with the glucocorticoids. The prototypical example is cortisol. It binds to and activates glucocorticoid receptors, abbreviated as GR, that is glucocorticoids. Now, mineralocorticoids, the main one is aldosterone, and the receptor is mineralocorticoid receptor, but there are other mineralocorticoids, other hormones that can activate the mineralocorticoid receptors. Usually, this is under pathophysiological conditions, and one example here is 11 deoxycorticosteroids, which is precursor in some of the steroid pathways. We are going to look at when these hormones builds up the typical. This is because an enzyme's defect that we will take about later, or sometimes there is hormone secreting tumors that make within the human. So the next one is progesterone, that is represented by PR. The common example of progesterones are progestins, such as it is also known as progesterones. The next one is androgens. The common examples of androgens are DHEA, androstenedione, testosterone, and dihydrotestosterone. It is represented by AR, means androgens. The last one is estrogens. The common examples of estrogens are estrone, estradiol, estriol. This is represented by ER. So, all the hormones are located at the adrenal cortex, gonads, and placenta within the mitochondrial positions, and all these hormones are synthesized from cholesterol. Means the basic precursor for the synthesis of these hormones are cholesterol. Let's see the steroid hormone synthesis. Steroids hormones are lipids molecule derived from a common cholesterol precursor, that is cholesterol carbon 27. Means the total number of carbon atom within the basic structures of the cholesterol is carbon 27. There are four major classes of steroid hormones: progestins, androgens, estrogens, and corticoids, which contain carbon 21, carbon 19, carbon 18, and other carbon 21, respectively. Steroid hormones are synthesized by dehydrogenases and cytochrome P450 enzymes, which catalyzed hydroxylations and dehydroxylation oxidation reactions. Eukaryotic cytochromes P450 are membrane bound enzymes expressed in either the inner mitochondrial or endoplasmic reticulum membranes of steroid synthesizing tissues. A common and important rate limiting step for the synthesis of all steroid hormones is the cleavage of side chain from cholesterol carbon 27 to shield pregnenolone, that is carbon 21. The common branch point for the synthesis of progestins, corticoids, androgens, and hence estrogens means, first of all, the pregnenolone is synthesized from the cholesterol molecule by the cleavage of side chain of the cholesterol. And after that, all other hormones that are progestins, corticoids, androgens, and estrogens are derived or synthesized from the pregnenolone, that is carbon 21. The expressions of the side chain cleavage enzymes, that is cytochrome, which is used for the cleavage of the side chain of the cholesterol, is 450 SCC, that is also expressed as site P450 SCC, which converts cholesterol to pregnenolone, is one of the unique features of steroidogenic cells that participate in the de novo steroid synthesis.